Welcome to this peaceful journey through the fascinating world of DNA and genetics. As you settle into comfort and relaxation, allow yourself to absorb the wonders of the molecular machinery that makes life possible. Close your eyes, breathe deeply, and let these facts about the building blocks of life gently guide you into restful sleep. DNA, or deoxyribonucleic acid, is the molecule that carries the genetic instructions for all known living organisms. This remarkable molecule exists in every cell of your body, quietly directing the processes that keep you alive. The structure of DNA was first described in 1953 by James Watson and Francis Crick, building upon the crucial X-ray crystallography work of Rosalind Franklin. Their discovery revealed that DNA forms a double helix, resembling a twisted ladder where the rungs are made of paired chemical bases. Each human cell contains approximately 6 feet of DNA, yet this incredibly long molecule is packed so efficiently that it fits inside a nucleus that is only about 6 micrometers in diameter. If you were to unwind all the DNA in your body and stretch it out, it would reach from Earth to the Sun and back more than 300 times. This extraordinary compaction is achieved through a sophisticated packaging system involving proteins called histones, around which DNA wraps like thread on a spool. The 4 chemical bases that make up DNA are adenine, thymine, guanine, and cytosine, often abbreviated as A, T, G, and C. These bases pair in specific ways, with adenine always pairing with thymine and guanine always pairing with cytosine, forming the rungs of the DNA ladder. This complementary base pairing is the key to how DNA replicates itself and how genetic information is preserved and transmitted from one generation to the next. Your genome, the complete set of genetic instructions contained in your DNA, consists of approximately 3 billion base pairs. These base pairs are organized into genes, which are specific sequences of DNA that provide instructions for making proteins. Proteins are the workhorses of your cells, performing countless functions from building cellular structures to catalyzing chemical reactions. The human genome contains approximately 20,000 to 25,000 genes, though this number is still being refined as scientists continue to study our genetic makeup. Interestingly, genes make up only about 2% of your entire genome. The remaining 98% was once called junk DNA, but researchers now know that much of this non-coding DNA plays important regulatory roles, controlling when and how genes are expressed. Genetic information flows from DNA to RNA to protein, in a process called the central dogma of molecular biology. First, the information in a gene is transcribed into messenger RNA, a temporary copy that can travel from the nucleus to the cellular machinery that builds proteins. Then, this messenger RNA is translated into a specific sequence of amino acids, which fold into functional proteins. Each 3-letter sequence of bases in the RNA, called a codon, specifies one amino acid in the protein chain. There are 64 possible codons, but only 20 standard amino acids, which means the genetic code is redundant, with multiple codons often specifying the same amino acid. This redundancy provides a buffer against mutations, as some changes in DNA sequence may not alter the resulting protein. Mutations are changes in the DNA sequence that can occur through various mechanisms. Some mutations arise from errors during DNA replication, when cells divide and copy their genetic material. Despite sophisticated proofreading mechanisms, mistakes occasionally slip through, occurring at a rate of about 1 error per billion base pairs copied. Other mutations result from damage caused by environmental factors such as ultraviolet radiation from the sun, chemicals in tobacco smoke, or naturally occurring reactive molecules in your cells. Most mutations are neutral, having no significant effect on an organism's survival or reproduction. Some mutations are harmful, disrupting normal protein function and potentially causing disease. A small fraction of mutations are beneficial, providing advantages that natural selection can favor over generations. The accumulation of mutations over time is the raw material for evolution, driving the diversity of life on Earth. Every human inherits half of their DNA from their mother and half from their father. This inheritance occurs through specialized reproductive cells called gametes, eggs and sperm, which contain only 23 chromosomes each instead of the usual 46 found in other body cells. When an egg and sperm unite during fertilization, they create a new individual with a complete set of 46 chromosomes, 23 pairs in total. 22 of these pairs are autosomes, chromosomes that are essentially the same in males and females. The 23rd pair consists of the sex chromosomes, which determine biological sex. Females typically have 2 X chromosomes, while males have 1 X and 1 Y chromosome. The Y chromosome is much smaller than the X and contains far fewer genes, but it carries the crucial SRY gene that triggers male development in the embryo. Chromosomes are not randomly distributed during the formation of gametes. A process called meiosis ensures that each gamete receives 1 chromosome from each pair, but which specific chromosome is a matter of chance. Additionally, before chromosomes separate, they undergo crossing over, exchanging segments of DNA with their paired partners. This genetic recombination shuffles genetic information, ensuring that each gamete and therefore each offspring is genetically unique, except in the case of identical twins. Identical twins result from a single fertilized egg that splits into 2 embryos, creating 2 individuals with virtually identical DNA sequences. Fraternal twins, by contrast, develop from 2 separate eggs fertilized by 2 separate sperm, making them no more genetically similar than regular siblings born at different times. The study of twins has been invaluable in understanding the relative contributions of genetics and environment to human traits and behaviors. Some traits are strongly influenced by a single gene, following simple patterns of inheritance first described by Gregor Mendel in his pioneering work with pea plants in the 1860s. These Mendelian traits include conditions like cystic fibrosis, sickle cell disease, and Huntington's disease. However, most human characteristics, from height and weight to intelligence and personality, are polygenic, influenced by many genes acting together in complex ways. These complex traits also depend heavily on environmental factors, making it challenging to predict how genetic variations will manifest in any individual. Genetic variations between individuals are surprisingly common. On average, any 2 unrelated humans differ at about 1 base pair in every 1,000, meaning each person has approximately 3 million single nucleotide differences compared to any other person. These single nucleotide polymorphisms, or SNPs, are the most common type of genetic variation among people. Some SNPs have no noticeable effect, while others influence traits like eye color, blood type, or susceptibility to certain diseases. Understanding these variations is a major focus of modern genetics research, as scientists work to connect specific genetic differences with health outcomes and treatment responses. The field of pharmacogenomics studies how genetic variations affect individual responses to medications. Some people metabolize drugs quickly, requiring higher doses for therapeutic effect, while others process medications slowly, risking toxic side effects from standard doses. Genetic testing can help doctors predict how patients will respond to certain medications, enabling more personalized treatment approaches. This precision medicine represents a paradigm shift from the traditional one-size-fits-all approach to healthcare. Genetic testing has become increasingly accessible and affordable in recent years, allowing individuals to learn about their ancestry, health risks, and carrier status for various genetic conditions. Direct-to-consumer genetic testing companies analyze hundreds of thousands of SNPs across the genome, comparing an individual's DNA with reference populations to infer ancestral origins and genetic health risks. However, interpreting these results requires caution, as genetic risk factors do not guarantee disease development, and many conditions depend on complex interactions between multiple genes and environmental factors. Genetic counselors are healthcare professionals specially trained to help people understand genetic test results and make informed decisions about their health and family planning. They can explain the implications of genetic variations, assess disease risks, and discuss available options for prevention, monitoring, or treatment. Genetic counseling is particularly valuable for families with a history of genetic conditions, or for individuals considering genetic testing. The ethical implications of genetic testing and genetic information are profound and continue to evolve. Questions about privacy, discrimination, and the use of genetic data raise important societal concerns. In the United States, the Genetic Information Non-Discrimination Act of 2008 provides some protections, prohibiting discrimination based on genetic information in health insurance and employment. However, these protections do not extend to all contexts, such as life insurance or long-term care insurance, and regulations vary internationally. Gene expression, the process by which genetic information is used to create functional products like proteins, is tightly regulated in cells. Not all genes are active all the time. Cells can turn genes on or off in response to developmental signals, environmental conditions, or cellular needs. This regulation occurs through various mechanisms, including transcription factors that bind to specific DNA sequences near genes, chemical modifications to DNA and histones that affect gene accessibility, and small RNA molecules that can block gene expression. Epigenetics is the study of changes in gene expression that do not involve alterations to the DNA sequence itself. Epigenetic modifications, such as the addition of methyl groups to DNA bases or the addition of various chemical tags to histone proteins, can persist through cell divisions and sometimes even across generations. These modifications can be influenced by environmental factors such as diet, stress, and exposure to toxins, providing a mechanism through which experiences and environmental conditions can affect gene expression and potentially health outcomes. The concept of epigenetic inheritance challenges the traditional view that inheritance is solely determined by DNA sequence. Research has shown that environmental exposures in parents or even grandparents can influence gene expression patterns in offspring through epigenetic mechanisms. Studies in model organisms have demonstrated that nutritional conditions, stress, and toxin exposure can have effects that persist for multiple generations. While the extent and significance of epigenetic inheritance in humans remain active areas of research, these findings highlight the complex interplay between genetics and environment in shaping traits and health. DNA methylation is one of the most studied epigenetic modifications. This process involves adding a methyl group to cytosine bases, particularly when they occur before guanine bases in the DNA sequence. Methylation typically reduces gene expression, effectively silencing genes without changing their underlying sequence. Patterns of DNA methylation change throughout life and can be influenced by age, lifestyle, and environmental exposures. Scientists are investigating whether methylation patterns can serve as biomarkers for disease risk or biological aging. Stem cells possess the remarkable ability to divide and differentiate into various specialized cell types. Embryonic stem cells, derived from early-stage embryos, are pluripotent, meaning they can become any cell type in the body. Adult stem cells, found in various tissues, are more limited in their potential, but still play crucial roles in tissue maintenance and repair. The discovery of methods to reprogram adult cells into induced pluripotent stem cells has revolutionized stem cell research, providing new avenues for studying development and disease, while avoiding some ethical concerns associated with embryonic stem cells. Understanding how genes control cell differentiation, the process by which stem cells become specialized, is a major focus of developmental biology. As cells differentiate, specific sets of genes are activated, while others are permanently silenced, determining the cell's ultimate identity and function. This process is guided by complex networks of transcription factors and signaling molecules that communicate between cells, ensuring that tissues and organs develop properly. Disruptions to these developmental programs can result in birth defects or contribute to cancer, when cells escape normal growth controls and differentiate abnormally. Cancer is fundamentally a genetic disease at the cellular level, arising from accumulating mutations that disrupt normal cell growth and division controls. Cancer cells typically harbor mutations in oncogenes, genes that promote cell growth and division, and tumor suppressor genes, which normally restrain cell proliferation or promote cell death when damage occurs. It usually takes multiple mutations accumulating over time for a cell to become fully cancerous, which helps explain why cancer incidence increases with age. Inherited mutations in certain genes can dramatically increase cancer risk, as in the case of BRCA1 and BRCA2 mutations that greatly elevate the likelihood of breast and ovarian cancer. However, most cancers result primarily from mutations acquired during a person's lifetime, rather than inherited variations. Understanding the genetic basis of cancer has led to targeted therapies that specifically attack cancer cells based on their particular genetic alterations. For example, some breast cancers overexpress the HER2 protein due to amplification of the HER2 gene, and drugs like trastuzumab specifically target this protein. Similarly, some leukemias result from specific chromosomal translocations that can be targeted with specialized medications. This precision oncology approach represents a major advance over traditional chemotherapy, which affects rapidly dividing cells indiscriminately. Genetic engineering and gene editing technologies have advanced rapidly in recent years, offering unprecedented abilities to modify DNA sequences in living organisms. The CRISPR-Cas9 system, adapted from a bacterial immune system, has revolutionized gene editing by providing a relatively simple, precise, and affordable method for making specific changes to genomic DNA. This technology has enormous potential for basic research, agriculture, and medicine. Scientists are exploring using CRISPR to correct genetic mutations that cause disease, modify crops for improved nutrition or climate resilience, and even control disease-carrying insect populations. The first human clinical trials using CRISPR to treat genetic diseases began in 2019, marking a new era in genetic medicine. However, gene editing also raises significant ethical questions, particularly regarding heritable genetic modifications that would be passed to future generations. In 2018, a Chinese scientist announced the birth of twin girls whose genomes he had edited as embryos, sparking international controversy and calls for strict regulation of germline editing. Most scientists and ethicists agree that while somatic gene therapy, which affects only the treated individual, is acceptable for serious diseases, germline editing that affects future generations should proceed with extreme caution, if at all until safety and ethical concerns are thoroughly addressed. The Human Genome Project, completed in 2003, was an international research effort that mapped and sequenced all the DNA base pairs in the human genome. This monumental achievement took 13 years and cost approximately $3 billion. Today, an individual's genome can be sequenced in less than a day for under $1,000, demonstrating the breathtaking pace of technological advancement in genomics. The dramatically reduced cost and time required for genome sequencing has opened new possibilities for research and personalized medicine. Comparative genomics, the study of similarities and differences in genome sequences across species, has provided profound insights into evolution and biology. By comparing human genomes with those of other organisms, scientists can identify conserved sequences that have remained largely unchanged over millions of years of evolution, suggesting they perform critical functions. Surprisingly, humans share about 99% of their DNA sequence with chimpanzees, our closest living relatives, and even share about 60% with fruit flies. These comparisons help identify uniquely human genes and genetic changes that contributed to human evolution. The concept of the molecular clock uses the rate of genetic mutations to estimate when species diverged from common ancestors. Because mutations accumulate at relatively steady rates over long time periods, comparing the number of genetic differences between species provides a genetic timestamp for evolutionary events. This molecular dating has helped refine the evolutionary tree of life, revealing relationships between organisms and timing of divergence, events that fossil evidence alone could not determine. Horizontal gene transfer, the movement of genetic material between organisms outside of traditional parent-to-offspring inheritance, is common in bacteria and has played a significant role in bacterial evolution. This process allows bacteria to rapidly acquire new capabilities, such as antibiotic resistance, which poses serious challenges for medicine and public health. While horizontal gene transfer is rare in complex organisms like animals, its occurrence in bacteria demonstrates that genetic information can flow through populations in ways beyond simple inheritance. The discovery of transposable elements, DNA sequences that can move around within the genome, challenge the view of genomes as static blueprints. These jumping genes, first discovered by Barbara McClintock in corn plants in the 1940s, make up nearly half of the human genome. Most transposable elements in humans are no longer active, but their past movements have contributed to genetic diversity and evolution. Some genetic diseases result from new insertions of active transposable elements, disrupting important genes. The mitochondria, the energy-producing organelles in your cells, contain their own DNA separate from the nuclear genome. Mitochondrial DNA is circular, much smaller than nuclear DNA, and is inherited almost exclusively from the mother. This maternal inheritance of mitochondrial DNA has made it useful for tracing maternal lineages and studying human migration patterns throughout history. Mutations in mitochondrial DNA can cause diseases affecting energy metabolism, often manifesting in tissues with high energy demands like muscle and brain. Population genetics studies how genetic variation is distributed within and between populations, and how this variation changes over time. Genetic drift, the random fluctuation of gene frequencies, has stronger effects in small populations and can lead to the loss or fixation of alleles regardless of their effects on fitness. Natural selection, by contrast, increases the frequency of beneficial alleles and decreases harmful ones, though its effects depend on population size, mutation rates, and environmental conditions. The founder effect occurs when a small group establishes a new population, carrying only a subset of the genetic variation from the original population, which can lead to different allele frequencies in the new population. Human evolution has been shaped by various selective pressures over millions of years. Some genetic adaptations are relatively recent in evolutionary terms, having emerged within the past 10,000 years. For example, lactase persistence, the ability to digest milk sugar into adulthood, evolved independently in several populations that practiced dairy farming, demonstrating how cultural practices can create selective pressures that shape human genetics. Similarly, genetic adaptations to high-altitude environments have evolved in Tibetan and Andean populations, allowing them to thrive in low-oxygen conditions that would cause severe problems for people from low-altitude populations. The neutral theory of molecular evolution, proposed by Madhu Kimura in 1968, suggests that much genetic variation at the molecular level is selectively neutral, neither beneficial nor harmful. According to this theory, most genetic changes that become fixed in populations do so through random genetic drift rather than natural selection. This perspective does not deny the importance of natural selection in shaping adaptations, but it emphasizes that many genetic differences between individuals and species may have little functional significance. Ancient DNA research has opened remarkable windows into the past, allowing scientists to extract and sequence DNA from fossils and archaeological remains. This work has revealed that modern humans interbred with Neanderthals and another archaic human group called Denisovans, with most non-African humans carrying 1 to 2% Neanderthal DNA in their genomes. These discoveries have fundamentally changed our understanding of human evolution, revealing a more complex picture of interbreeding and replacement than previously thought. Analysis of ancient DNA from crop plants and domesticated animals has illuminated the origins and spread of agriculture, one of the most transformative developments in human history. Genetic engineering in agriculture has created crops with enhanced traits such as pest resistance, herbicide tolerance, improved nutrition, and better yield. Genetically modified organisms, or GMOs, have been controversial, with debates about their safety, environmental impact, and ethical implications. While scientific consensus holds that currently approved GMO crops are safe for consumption and have not caused documented harm to human health, concerns about biodiversity, pesticide use, and corporate control of agriculture continue to fuel discussions about agricultural biotechnology. Gene drives are genetic engineering systems designed to spread specific genes through populations more rapidly than normal inheritance would allow. These technologies could potentially control disease-carrying mosquitoes or invasive species, but they also raise serious ecological concerns about unintended consequences and the irreversibility of releasing self-propagating genetic modifications into the environment. The international scientific community is actively debating appropriate governance frameworks for gene drive research and potential applications. Synthetic biology aims to design and construct new biological systems and organisms with novel functions, treating biological components as engineering parts that can be mixed and matched to create desired outcomes. This field has produced bacteria engineered to produce medications, biofuels, and industrial chemicals, as well as efforts to create minimal genomes. stripped-down versions of bacterial genomes containing only essential genes. Some researchers are working toward creating entirely synthetic cells from scratch, raising profound questions about the nature of life and our ability and right to create it. The genetic code, the system by which nucleotide triplets specify amino acids, is nearly universal across all life on Earth, from bacteria to humans. This universality provides strong evidence for the common ancestry of all living things and enables genetic engineering across species boundaries, allowing human genes to function in bacteria and vice versa. However, some organisms use slightly modified genetic codes, and scientists have even created synthetic organisms with expanded genetic codes that incorporate unnatural amino acids, opening new possibilities for protein engineering. RNA plays many roles beyond serving as a messenger between DNA and proteins. Ribosomal RNA is a crucial component of ribosomes, the molecular machines that synthesize proteins. Transfer RNA molecules carry amino acids to the ribosome and decode the genetic message. Many other types of RNA molecules perform regulatory functions, controlling gene expression and affecting various cellular processes. The discovery of ribozymes, RNA molecules with catalytic activity, led to the RNA world hypothesis, which proposes that early life may have relied on RNA as both information carrier and catalyst before the evolution of DNA and proteins. MicroRNAs are small RNA molecules that regulate gene expression by binding to messenger RNAs and preventing their translation into proteins. These tiny regulators play crucial roles in development, cellular differentiation, and disease. Changes in microRNA expression have been associated with various cancers and other disorders, and some researchers are exploring microRNAs as therapeutic targets or disease biomarkers. Long non-coding RNAs are another class of regulatory RNA molecules that do not encode proteins, but influence gene expression through various mechanisms, demonstrating that the functional landscape of the genome extends far beyond protein-coding genes. Telomeres are protective caps on the ends of chromosomes, consisting of repetitive DNA sequences and associated proteins. Each time a cell divides, telomeres become slightly shorter. When telomeres become too short, cells stop dividing or die, which is thought to contribute to aging and age-related diseases. The enzyme telomerase can rebuild telomeres, and it is active in stem cells and unfortunately also in many cancer cells, contributing to their ability to divide indefinitely. Understanding telomere biology has implications for aging research and cancer treatment. The p53 gene, often called the guardian of the genome, encodes a protein that helps protect cells from becoming cancerous. When DNA damage occurs, p53 can halt cell division to allow time for repair or trigger cell death if damage is too severe to fix. Mutations in p53 are among the most common genetic changes in human cancers, found in more than half of all tumors. The critical importance of p53 in preventing cancer highlights how genetic safeguards normally protect us from malignancy, and how their failure can lead to disease. Chromosomal disorders result from abnormalities in chromosome number or structure. Down syndrome, the most common chromosomal condition, results from having 3 copies of chromosome 21 instead of the usual 2. Turner syndrome affects females who have only one X chromosome instead of 2. Klinefelter syndrome affects males who have an extra X chromosome. These conditions demonstrate how the precise balance of genetic material is crucial for normal development. Many chromosomal abnormalities are not compatible with life and result in early miscarriage, highlighting the delicate balance required for human development. Genetic imprinting is a phenomenon where certain genes are expressed differently depending on whether they were inherited from the mother or father. Imprinted genes are marked with epigenetic modifications during gamete formation, and these marks determine whether the gene will be active or silent. Disorders of imprinting, such as Prader-Willi syndrome and Angelman syndrome, can result from deletions of the same chromosomal region. But the specific symptoms depend on whether the deletion came from the mother or father, due to differential imprinting of genes in that region. The discovery of genetic imprinting challenged the assumption that maternal and paternal genetic contributions are equivalent. DNA damage occurs constantly in cells from both external sources like radiation and chemicals, and internal sources like reactive oxygen species produced during normal metabolism. Cells have sophisticated DNA repair mechanisms that fix most damage before it becomes permanent. Different repair pathways handle different types of damage, from simple base modifications to complex breaks in both DNA strands. Defects in DNA repair genes can dramatically increase mutation rates and disease risk. For example, mutations in genes involved in repairing mismatched base pairs cause Lynch syndrome, which greatly increases colorectal cancer risk. Understanding DNA repair has implications for cancer prevention and treatment, as some cancer therapies work by overwhelming cancer cells with DNA damage they cannot repair. Genetic mosaicism occurs when an individual has two or more genetically distinct cell populations, arising from mutations that occur after fertilization. Somatic mosaicism, where the mutation is present in some body cells but not in reproductive cells, is common and usually harmless. However, mosaic mutations can cause disease if they affect critical genes and occur early enough in development to affect many cells. Germline mosaicism, where mutations are present in reproductive cells, can result in parents without a genetic condition having multiple affected children, complicating genetic counseling. DNA profiling, also called DNA fingerprinting, uses variations in specific regions of DNA to identify individuals with extremely high probability. This technology has revolutionized forensic science, paternity testing, and identification of human remains. Modern DNA profiling typically analyzes short tandem repeats, sequences of 2 to 6 base pairs that are repeated varying numbers of times in different individuals. The probability that two unrelated individuals will have the same profile at multiple DNA markers is astronomically small, making DNA profiling highly reliable for identification purposes. Mitochondrial DNA analysis is particularly useful in forensic applications because mitochondrial DNA is present in many copies per cell, making it more likely to be recovered from degraded samples. And because it is maternally inherited, allowing matching to maternal relatives when direct comparison samples are unavailable. Ancient DNA analysis and forensic applications both benefit from these properties of mitochondrial DNA. The Y chromosome, passed from father to son with little change across generations, is useful for tracing paternal lineages and studying human migration patterns. Genetic genealogy, combining DNA testing with traditional genealogical records, has become popular for tracing ancestry and finding relatives. Law enforcement has also begun using genetic genealogy databases to identify suspects in criminal cases by finding relatives who have uploaded their DNA profiles to public databases. Raising important privacy concerns about the use of genetic information beyond its originally intended purposes. Pharmacogenomics promises to revolutionize medicine by enabling truly personalized treatment based on individual genetic profiles. Already, genetic testing guides dosing decisions for drugs like warfarin, a blood thinner with a narrow therapeutic window, and helps predict which cancer patients will respond to specific targeted therapies. As our understanding of how genetic variations affect drug responses grows, routine genetic screening before prescribing medications may become standard practice, reducing adverse drug reactions and improving treatment effectiveness. The gut microbiome, the community of microorganisms living in your digestive system, has its own collective genome called the metagenome, containing millions of genes and far outnumbering human genes. These microbial genes influence digestion, metabolism, immune function, and even behavior. The composition of your microbiome is influenced by genetics, but also significantly shaped by diet, environment, and antibiotic use. Research into the human microbiome and its genetic makeup is revealing the profound ways in which we are not just individual organisms, but complex ecosystems. Xenotransplantation, the transplantation of organs from animals to humans, has been pursued as a potential solution to organ shortage. Pigs are considered the most promising donors due to their similar organ sizes and physiology. However, rejection and cross-species viral transmission have been major obstacles. Recent advances in genetic engineering, including using CRISPR to remove porcine endogenous retroviruses from pig genomes and modify pig tissues to be more compatible with human immune systems, have brought xenotransplantation closer to clinical reality. The first successful transplant of a genetically modified pig heart into a human patient occurred in 2022, marking a milestone in this field. Genetic privacy concerns are growing as genetic testing becomes more common and databases of genetic information expand. Your genetic information is uniquely identifying and reveals information not just about you, but also about your blood relatives who share portions of your DNA. Questions about who should have access to genetic information, how it should be stored and protected, and what purposes it can be used for are increasingly important. The potential for genetic discrimination in employment, insurance, or other contexts remains a concern despite legal protections in many jurisdictions. Precision medicine aims to tailor medical treatment to individual characteristics, including genetic makeup, lifestyle, and environment. This approach recognizes that people respond differently to treatments, and that a one-size-fits-all approach often leads to suboptimal outcomes. Genetic information is just one component of precision medicine, which also incorporates data from imaging, laboratory tests, and patient-reported outcomes to provide more individualized care. As data science and artificial intelligence improve our ability to analyze complex medical information, precision medicine is expected to become increasingly sophisticated and widely implemented. Gene therapy, the introduction of genetic material into cells to treat disease, has achieved remarkable successes after decades of research and setbacks. Early gene therapy trials in the 1990s faced challenges, including immune reactions and insertional mutagenesis, where viral vectors used to deliver therapeutic genes inserted themselves into harmful locations in the genome. Modern gene therapy approaches use improved vectors and delivery methods, and several gene therapies have been approved for treating genetic diseases including inherited blindness, severe combined immunodeficiency, and spinal muscular atrophy. These treatments represent hope for patients with previously untreatable genetic conditions. Regenerative medicine seeks to repair or replace damaged tissues and organs, often utilizing stem cells and understanding of developmental genetics. Tissue engineering combines cells, biomaterial scaffolds, and growth factors to create functional tissues. While creating complex organs remains a major challenge, scientists have successfully engineered simpler tissues like skin and cartilage. Understanding the genetic programs that guide organ development during embryonic growth is crucial for regenerative medicine efforts to recapitulate these processes for therapeutic purposes. Systems biology takes a holistic approach to understanding biological systems, recognizing that genes do not function in isolation, but as parts of complex networks. Gene regulatory networks describe how genes influence each other's expression, creating feedback loops and interconnections that determine cellular behavior. Metabolic networks map the chemical reactions in cells and how they are interconnected. Analyzing these networks requires computational approaches and reveals emergent properties that cannot be understood by studying individual genes alone. Systems biology is particularly relevant for understanding complex diseases like cancer, diabetes, and neurological disorders that involve dysfunction in multiple interconnected pathways. Evolutionary developmental biology, or EVO, investigates how changes in developmental genes and processes lead to evolutionary changes in body form. This field has revealed that many diverse organisms use similar genetic toolkits of developmental control genes, with differences in how and when these genes are expressed accounting for much of the diversity in animal form. Hox genes, for example, specify body segment identity in organisms from flies to humans, despite these organisms having vastly different body plans. Understanding how genetic changes alter development illuminates how evolution produces biological diversity. As you continue to relax and drift toward sleep, consider how the instructions encoded in DNA molecules orchestrate the incredible complexity of life. From the precise pairing of chemical bases to the intricate regulation of thousands of genes, from the inheritance of traits across generations to the evolution of species over millions of years. Genetics touches every aspect of biology. The DNA in your cells connects you to every human who has ever lived, to all organisms on Earth through common ancestry, and to future generations who will carry forward the genetic legacy you pass on. Rest peacefully knowing that countless molecular processes, guided by genetic instructions refined over billions of years of evolution, are working harmoniously to maintain your health and well-being. Chromosomes condense dramatically during cell division, becoming visible as distinct X-shaped structures under a microscope. During most of a cell's life, however, DNA exists in a less condensed form called chromatin, a complex of DNA and proteins that allows the cellular machinery to access genes as needed. The level of chromatin condensation influences gene accessibility, with tightly packed chromatin generally associated with gene silencing, and loosely packed chromatin associated with active gene expression. Histone modifications, including acetylation, methylation, and phosphorylation, influence chromatin structure and play crucial roles in regulating gene expression. Chromatin remodeling complexes are protein machines that can reposition or remove histones, making DNA more or less accessible to transcription factors and other regulatory proteins. These epigenetic mechanisms provide a flexible way to control gene expression without altering the underlying DNA sequence. The nucleosome, the basic unit of chromatin structure, consists of DNA wrapped around a core of 8 histone proteins. Approximately 147 base pairs of DNA wind around each histone octamer, creating a bead-on a string appearance when chromatin is visualized at low condensation levels. Nucleosomes are regularly spaced along DNA, though their positions can shift in response to cellular signals. The linker DNA between nucleosomes varies in length, and linker histone proteins help further compact the chromatin structure. During DNA replication, which occurs before cell division, specialized enzymes called DNA polymerases synthesize new DNA strands complementary to existing template strands. Replication begins at specific sites called origins of replication, where proteins recognize particular DNA sequences and initiate the unwinding of the double helix. In humans, replication begins simultaneously at thousands of origins across all chromosomes, ensuring the entire genome can be copied in a reasonable time. Replication proceeds bidirectionally from each origin, with replication forks moving in opposite directions along the DNA. DNA polymerase can only add nucleotides to an existing strand, requiring primer sequences to initiate synthesis. RNA primase synthesizes short RNA primers that DNA polymerase extends. Leading strand synthesis proceeds continuously in the same direction as the replication fork movement, while lagging strand synthesis occurs discontinuously in short segments called Okazaki fragments. DNA ligase later joins these fragments to create a continuous strand. Proofreading mechanisms during replication ensure remarkable accuracy. DNA polymerase has exonuclease activity that allows it to remove incorrectly paired nucleotides and replace them with correct ones. Despite this proofreading, errors occasionally persist, contributing to the mutation rate. Mismatch repair systems provide an additional layer of quality control. Scanning newly synthesized DNA for errors missed by polymerase proofreading and correcting them before the next round of replication. Telomeres present a special challenge for DNA replication because DNA polymerase cannot fully replicate the ends of linear chromosomes. This end replication problem would cause chromosomes to shorten with each cell division, if not for telomerase, an enzyme that adds repetitive sequences to chromosome ends. Most adult cells have low or absent telomerase activity, leading to progressive telomere shortening with each division. This shortening contributes to cellular aging and the limited replicative capacity of most cells, a phenomenon called the Hayflick limit after the scientist who first described it. Cell division occurs through two main processes, mitosis and meiosis. Mitosis produces two identical daughter cells from a single parent cell, maintaining the same chromosome number. This process is essential for growth, tissue repair, and asexual reproduction. Meiosis, by contrast, produces four non-identical gametes, each with half the chromosome number of the parent cell. This reduction division is essential for sexual reproduction, ensuring that fertilization restores the normal chromosome number rather than doubling it each generation. Mitosis proceeds through several distinct phases. During prophase, chromosomes condense and become visible, while the mitotic spindle begins to form. Prometophase follows, with the nuclear envelope breaking down and spindle fibers attaching to chromosomes at specialized structures called kinetochores located at the centromeres. In metaphase, chromosomes align at the cell's equator, forming the metaphase plate. Anaphase begins when sister chromatids separate and move toward opposite poles of the cell, pulled by shortening spindle fibers. Finally, during telophase, nuclear envelopes reform around the separated chromosome sets, and the chromosomes begin to decondense. Cytokinesis, the physical division of the cell, typically occurs during telophase, creating two separate daughter cells. Meiosis involves two successive divisions, meiosis I and meiosis II, each with their own prophase, metaphase, anaphase, and telophase stages. Meiosis I is the reduction division, separating homologous chromosomes so that each daughter cell receives only one chromosome from each pair. Prophase I is particularly lengthy and important, as this is when crossing over occurs, exchanging genetic material between homologous chromosomes. This recombination shuffles genetic information, creating new combinations of alleles that contribute to genetic diversity. Meiosis II resembles mitosis, separating sister chromatids to produce 4 haploid cells from the 2 cells resulting from meiosis I. Errors in cell division can have serious consequences. Non-disjunction, the failure of chromosomes or chromatids to separate properly, results in gametes with abnormal chromosome numbers. If such gametes participate in fertilization, the resulting embryo will have aneuploidy, an abnormal number of chromosomes. Trisomy, having 3 copies of a chromosome instead of 2, is compatible with life for only a few chromosomes, most notably chromosomes 13, 18, and 21. Most aneuploidies are lethal and result in early miscarriage. Monosomy, having only 1 copy of a chromosome, is typically even more detrimental than trisomy, with monosomy of the X chromosome being the only monosomy compatible with survival. The cell cycle, the series of events that leads to cell division, is tightly regulated by cyclins and cyclin-dependent kinases that control progression through different phases. The cycle consists of interphase, when the cell grows and replicates its DNA, and the mitotic phase, when division occurs. Interphase itself has 3 stages: G1 phase for cell growth, S phase when DNA synthesis occurs, and G2 phase for preparation for division. Checkpoints at transitions between phases ensure that conditions are appropriate before proceeding. The G1 checkpoint verifies that the cell is large enough and conditions are favorable for division. The G2 checkpoint ensures DNA has been completely and accurately replicated. The metaphase checkpoint, also called the spindle checkpoint, confirms that all chromosomes are properly attached to spindle fibers before allowing separation. Cancer often involves mutations that disrupt cell cycle control, allowing cells to bypass these checkpoints and divide uncontrollably. The study of cell cycle regulation has been crucial for understanding cancer and developing treatments. Many cancer therapies target rapidly dividing cells, exploiting the fact that cancer cells divide more frequently than most normal cells. However, this approach also affects normal rapidly dividing cells, such as those in hair follicles and the digestive system lining, causing common chemotherapy side effects like hair loss and nausea. Newer targeted therapies aim to specifically attack cancer cells based on their unique genetic characteristics, sparing normal cells and reducing side effects. Apoptosis, or programmed cell death, is a normal process essential for development and tissue maintenance. During embryonic development, apoptosis sculpts structures by removing cells that are no longer needed, such as the tissue between developing fingers and toes. In adults, apoptosis removes damaged or potentially dangerous cells, providing protection against cancer. Cells that have accumulated too much DNA damage or that have been infected by viruses typically undergo apoptosis, unless these safety mechanisms are disrupted. Evasion of apoptosis is a hallmark of cancer cells, allowing them to survive despite damage or abnormalities that would trigger death in normal cells. The p53 protein plays a central role in triggering apoptosis in response to DNA damage or cellular stress. When activated, p53 can induce expression of genes that halt the cell cycle, attempt DNA repair, or if damage is too severe, initiate apoptosis. Given its crucial role in protecting against cancer, p53 has been called the guardian of the genome. Mutations that inactivate p53 are found in more than half of human cancers, highlighting how loss of this tumor suppressor removes a critical barrier to malignancy. Researchers are exploring ways to restore p53 function in cancer cells, as a therapeutic strategy. DNA damage response pathways coordinate cell cycle arrest, DNA repair, and apoptosis in response to various types of DNA damage. Different types of damage activate specific response pathways. Double-strand breaks, among the most serious forms of damage, are detected by sensor proteins that recruit repair machinery and signal kinases to halt the cell cycle. Failure to repair double-strand breaks can lead to chromosomal rearrangements, deletions, or other changes that contribute to cancer development. Understanding DNA damage responses has implications for both cancer prevention and treatment, as some cancer therapies work by inducing irreparable DNA damage in cancer cells. Base excision repair corrects small-scale damage affecting individual bases, such as oxidative damage or deamination. This repair pathway involves specialized enzymes called glycosylases that recognize and remove damaged bases, creating a basic sites. Other enzymes then process these sites, inserting correct bases to restore the original sequence. Nucleotide excision repair handles bulkier lesions that distort the DNA helix, such as those caused by ultraviolet light exposure. This pathway removes an entire segment of damaged DNA and resynthesizes the correct sequence, using the undamaged strand as a template. Xeroderma pigmentosum, a rare genetic disorder involving defective nucleotide excision repair, causes extreme sensitivity to sunlight and dramatically increased skin cancer risk. Demonstrating the importance of this repair pathway. Homologous recombination and non-homologous end joining are two major pathways for repairing double-strand breaks. Homologous recombination uses the intact sister chromatid or homologous chromosome as a template to accurately repair the break. Predominating during and immediately after DNA replication when such templates are available. Non-homologous end joining directly ligates broken DNA ends without requiring a template. Operating throughout the cell cycle but being more error-prone because it may cause small deletions or insertions at the repair site. Mutations in genes involved in homologous recombination, including BRCA1 and BRCA2, greatly increase cancer risk because cells cannot accurately repair double-strand breaks, leading to accumulation of chromosomal abnormalities. The BRCA genes, originally identified through studies of families with high rates of breast and ovarian cancer, encode proteins involved in homologous recombination repair. Women with harmful mutations in BRCA1 or BRCA2 have up to 70% lifetime risk of breast cancer and up to 40% risk of ovarian cancer, compared to much lower risks in the general population. Men with BRCA mutations also have increased risks of breast cancer and prostate cancer. Genetic testing for BRCA mutation allows at-risk individuals to make informed decisions about prevention and screening strategies. Some women with BRCA mutations choose preventive surgery to remove breasts or ovaries, dramatically reducing their cancer risk. DNA methylation patterns are established and maintained by DNA methyltransferase enzymes. De novo methyltransferases establish new methylation patterns, while maintenance methyltransferases copy existing patterns to newly synthesized DNA strands during replication, preserving epigenetic information through cell divisions. Aberrant DNA methylation is common in cancer, with global hypomethylation often accompanied by hypermethylation of specific gene promoters. Hypermethylation of tumor suppressor gene promoters effectively silences these protective genes without requiring mutations, contributing to cancer development. Drugs that inhibit DNA methyltransferases have been developed as cancer treatments, aiming to restore expression of silenced tumor suppressors. Histone acetylation generally activates gene expression by neutralizing positive charges on histones, loosening their interaction with negatively charged DNA. Histone acetyltransferases add acetyl groups to lysine residues on histone tails, while histone deacetylases remove them. The balance between these opposing activities helps determine chromatin structure and gene expression levels. Histone deacetylase inhibitors have been developed as cancer treatments, with several approved for specific blood cancers. These drugs can reactivate silenced genes and induce cancer cell death through various mechanisms. Histone methylation can either activate or repress gene expression, depending on which specific lysine residues are modified and how many methyl groups are added. Trimethylation of histone 3 at lysine 4, for example, is associated with active gene expression, while trimethylation of histone 3 at lysine 9 or lysine 27 is associated with gene silencing. Histone methyltransferases and demethylases regulate these modifications, and their dysregulation has been implicated in cancer and other diseases. Some cancer drugs target specific histone methyltransferases or demethylases, aiming to correct aberrant gene expression patterns. The histone code hypothesis proposes that combinations of histone modifications constitute a code that is read by other proteins to determine chromatin structure and gene expression. Specific modifications recruit particular proteins that recognize and bind to modified histones, bringing additional factors that further modify chromatin or influence transcription. This complex regulatory system allows precise control of gene expression in response to developmental and environmental signals. Chromatin immunoprecipitation, or CHIP, is a technique used to identify which regions of the genome are associated with specific histone modifications or DNA-binding proteins. This method has been crucial for mapping the locations of various chromatin marks and transcription factor binding sites across entire genomes, revealing the regulatory landscape that controls gene expression. CHIP-seq, which combines chromatin immunoprecipitation with high-throughput DNA sequencing, has generated comprehensive maps of histone modifications and transcription factor occupancy for many cell types and organisms. Transcription factors are proteins that bind to specific DNA sequences and regulate gene expression. Some transcription factors are general, required for transcription of most genes, while others are specific, controlling particular sets of genes in response to developmental or environmental signals. Transcription factor binding sites are typically short DNA sequences, often 6 to 12 base pairs long, that occur in regulatory regions near genes. Combinations of transcription factors binding to multiple sites determine whether and how strongly a gene is expressed. The modular nature of gene regulation, with multiple binding sites for different factors, allows genes to respond to complex combinations of signals. Enhancers are regulatory DNA sequences that can increase gene expression levels, sometimes from great distances. Enhancers contain multiple transcription factor binding sites and can function regardless of their orientation or distance from the gene they regulate, sometimes located hundreds of thousands of base pairs away. DNA looping brings enhancers into physical proximity with gene promoters, allowing bound transcription factors to interact with the transcriptional machinery. Silencers function oppositely to enhancers, decreasing gene expression when bound by repressor proteins. Insulators are DNA sequences that block the effects of enhancers, preventing them from inappropriately activating nearby genes. The three-dimensional organization of chromatin in the nucleus plays important roles in gene regulation. Chromosomes occupy distinct territories in the nucleus, and genes are not randomly positioned, but rather organized in a way that reflects their regulatory relationships. Active genes tend to be located in less condensed chromatin regions toward the interior of the nucleus, while inactive genes are often found in condensed heterochromatin near the nuclear envelope. Chromosome conformation capture techniques have revealed that enhancers and their target genes are often brought together in three-dimensional space through DNA looping, even when separated by large linear distances. Topologically associating domains are regions of chromosomes that preferentially interact with themselves rather than with neighboring regions, creating distinct regulatory neighborhoods. Disruption of topological domain boundaries can lead to inappropriate gene activation by allowing enhancers to interact with wrong target genes, which has been implicated in developmental disorders and cancer. The study of three-dimensional genome organization has revealed that genome structure is not static but dynamic, changing during development and in response to environmental signals. Alternative splicing is a process that allows a single gene to produce multiple different protein variants by including or excluding different combinations of exons in the final messenger RNA. Most human genes undergo alternative splicing, greatly expanding the protein coding capacity of the genome beyond the number of genes it contains. Splicing is carried out by the spliciosome, a large complex of RNA and protein that recognizes specific sequences marking exon-intron boundaries. Splicing factors influence which exons are included or skipped, and their expression levels can vary between cell types and conditions, allowing tissue-specific or condition-specific protein variants. Mutations that affect splicing are increasingly recognized as important causes of genetic disease. Single nucleotide changes can create or destroy splice sites, cause exon skipping or inclusion, or activate cryptic splice sites, all of which can alter the resulting protein in harmful ways. Some genetic diseases result primarily from splicing defects, rather than changes to protein coding sequences. Therapeutic approaches targeting splicing, including antisense oligonucleotides that modify splicing patterns, have shown promise for treating certain genetic diseases. Spinal muscular atrophy, a severe neuromuscular disorder, can be treated with antisense oligonucleotides that alter splicing of the SMN2 gene to produce more functional protein, demonstrating the therapeutic potential of modulating splicing. RNA editing is another mechanism that can alter genetic information after transcription. Adenosine-twinosine editing, carried out by ADAR enzymes, changes specific adenosine bases in RNA to inosine, which is read as guanosine by the translation machinery. This editing can alter protein sequences, affect RNA splicing, or change regulatory regions of messenger RNAs. RNA editing is particularly common in the brain, where it fine-tunes the properties of neurotransmitter receptors and other proteins important for neuronal function. Dysregulation of RNA editing has been associated with neurological and psychiatric disorders. The epitranscriptome refers to chemical modifications of RNA bases beyond the standard 4 nucleotides. More than 100 different RNA modifications have been identified, with functions ranging from affecting RNA stability to influencing translation efficiency. N6-methyladenosine, the most abundant internal modification in messenger RNAs, affects RNA processing, localization, translation, and decay. Enzymes that add or remove RNA modifications regulate the epitranscriptome, and these modifications provide an additional layer of gene expression control. Research into RNA modifications and their functions is an active and rapidly evolving field. MicroRNA biogenesis begins with transcription of primary microRNA transcripts, which are processed in the nucleus to precursor microRNAs, and then further processed in the cytoplasm to mature microRNAs. The mature microRNA associates with proteins in the RNA-induced silencing complex, which uses the microRNA sequence to identify complementary messenger RNAs. Depending on the degree of complementarity, the target messenger RNA may be degraded, or its translation may be blocked. A single microRNA can regulate dozens to hundreds of different messenger RNAs, and individual messenger RNAs are often regulated by multiple microRNAs, creating complex regulatory networks. The discovery that microRNAs can be packaged into extracellular vesicles and transferred between cells has revealed intercellular communication functions for these small RNAs. Circulating microRNAs in blood can serve as biomarkers for various diseases, as different conditions are associated with characteristic microRNA profiles. The potential of microRNAs as therapeutic targets and as biomarkers continues to be explored in numerous clinical trials. Circular RNAs are a class of non-coding RNAs formed when the ends of a linear RNA molecule are joined together, creating a closed loop. These molecules are surprisingly abundant and stable, resisting degradation by exonucleases that normally break down linear RNAs. Some circular RNAs function as microRNA sponges, binding to and sequestering microRNAs to prevent them from regulating their normal targets. Other circular RNAs may have roles in regulating transcription or even producing peptides. The functions and significance of most circular RNAs remain to be fully understood, but their existence adds another layer to the complexity of gene regulation. CRISPR-Cas9 gene editing works by directing a Cas9 nuclease to a specific DNA sequence using a guide RNA molecule complementary to the target. The Cas9 enzyme cuts both strands of DNA at the targeted location, creating a double-strand break. Cells then repair this break using either non-homologous end joining, which often introduces small insertions or deletions that disrupt the gene, or homologous recombination if a repair template is provided, which can be used to insert specific sequences or correct mutations. CRISPR has been rapidly adopted for research because of its simplicity, efficiency, and versatility compared to previous gene editing methods. Base editing and prime editing are newer CRISPR-derived technologies that allow more precise genetic changes without creating double-strand breaks. Base editors convert one DNA base to another, for example changing a CG pair to a TA pair, without cutting the DNA backbone. Prime editors can insert, delete, or replace DNA sequences at target sites with even greater precision and flexibility. These technologies expand the capabilities of genome editing and may be particularly useful for therapeutic applications where precise correction of point mutations is desired. The off-target effects of CRISPR, where Cas9 cuts at unintended sites with similar sequences to the target, have been a concern for therapeutic applications. Researchers have developed high-fidelity Cas9 variants with reduced off-target activity and methods for detecting and minimizing unwanted edits. Delivery of CRISPR components to target cells and tissues remains a challenge, particularly for in vivo applications. Viral vectors, lipid nanoparticles, and other delivery methods are being developed and optimized for different therapeutic applications. Clinical trials using CRISPR for treating various genetic diseases and cancers are underway, with some showing promising early results. Inherited retinal diseases, sickle cell disease, and beta thalassemia are among the conditions being targeted in these trials. The first CRISPR therapy was approved in late 2023 for treating sickle cell disease and beta thalassemia, marking a milestone in the translation of gene editing from laboratory to clinic. These treatments work by editing hematopoietic stem cells ex vivo before transplanting them back into patients, avoiding some of the challenges of in vivo delivery. Somatic cell gene therapy modifies genes in body cells but not in reproductive cells, so changes are not inherited by offspring. Germline gene therapy would modify genes in reproductive cells or early embryos, creating heritable changes that would be passed to future generations. While somatic gene therapy for treating serious diseases is generally accepted, germline editing remains controversial due to ethical concerns about modifying human heredity, potential unintended consequences for future generations, and social justice issues regarding access and applications. International scientific and ethical bodies have called for broad societal dialogue before any clinical use of germline editing, and most countries have regulations prohibiting or strictly limiting such interventions. The rapid pace of technological advancement in genetics and genomics creates ongoing needs for updated ethical frameworks and policies. Issues such as genetic privacy, equitable access to genetic technologies, responsible use of gene editing, and the societal implications of enhanced understanding of human genetic variation require ongoing attention from scientists, ethicists, policymakers, and the public. Genetic literacy, understanding basic concepts of genetics and inheritance, is increasingly important as genetic information becomes more integrated into healthcare, and as genetic technologies affect more aspects of society. Ancient DNA research faces unique challenges because DNA degrades over time, breaking into short fragments and undergoing chemical modifications. Successful ancient DNA studies require careful contamination control to distinguish authentic ancient sequences from modern contamination. Cold, dry environments preserve DNA best, allowing successful extraction and analysis of DNA from specimens tens of thousands of years old. DNA from frozen mammoths, Neanderthal bones from cool caves, and preserved plant remains has provided invaluable insights into evolutionary history, past climates, and human migrations and interactions with other hominin species. Paleogenomics, the study of ancient genomes, has revealed surprising complexity in human evolutionary history. Modern humans, Neanderthals, and Denisovans all interbred to varying degrees, with non-African modern humans carrying Neanderthal DNA and some Asian and Oceanian populations carrying Denisovan DNA. These archaic human genes have influenced modern human traits, including immune system function, adaptation to high altitude, and skin characteristics. Some Neanderthal genes that were initially beneficial may now increase disease risk in modern environments, a phenomenon called genetic mismatch. Ancient pathogen DNA has revealed the evolutionary history of diseases that have shaped human populations. DNA from victims of historical plague outbreaks has traced the evolution and spread of Yersinia pestis, the bacterium that caused the Black Death and other devastating pandemics. Ancient tuberculosis DNA from archaeological remains has illuminated the origins and evolution of this persistent human pathogen. Understanding pathogen evolution through ancient DNA helps contextualize modern disease threats and informs public health strategies. Domestication of plants and animals involved genetic changes selected by humans over thousands of years. Comparing genomes of domesticated species with their wild relatives reveals the genes and genetic changes that underlie domesticated traits. Dogs domesticated from wolves show genetic changes affecting behavior, cognition, and morphology. Domesticated crops show genetic changes affecting seed size, plant architecture, and loss of seed dispersal mechanisms. Understanding the genetic basis of domestication has both historical interest and practical applications for crop improvement. The genetic diversity of crop plants has been reduced through domestication and modern breeding, creating vulnerability to pests, diseases, and environmental changes. Crop wild relatives retain genetic diversity that may include valuable traits for improving cultivated varieties. Gene banks preserve seeds and genetic material from diverse crop varieties and wild relatives, safeguarding this genetic diversity for future use. As climate change and emerging diseases threaten food security, this genetic diversity may prove crucial for developing resilient crop varieties. Synthetic genomics involves designing and constructing artificial genomes. Scientists have synthesized the complete genomes of bacteria and yeast, and work continues toward synthesizing more complex genomes. These efforts advance our understanding of genome function by revealing which genes are truly essential and how genomes are organized. Synthetic genomics also enables the creation of organisms with novel capabilities, such as bacteria programmed to produce pharmaceuticals, biofuels, or other valuable compounds. The ability to write DNA sequences de novo raises questions about the limits and ethics of human intervention in living systems. Genetic circuits, analogous to electronic circuits, are designed networks of genes and regulatory elements engineered to perform specific functions. These circuits can implement logic gates, oscillators, switches, and other computational functions within living cells. Applications include biosensors that detect environmental contaminants or disease markers, and therapeutic cells programmed to respond to disease signals. Synthetic biology aims to make biology more predictable and engineering-friendly, treating biological components as standardized parts that can be assembled into functional systems. Xenobiology explores the possibility of alternative genetic systems beyond the standard DNA-based life found on Earth. Researchers have expanded the genetic alphabet beyond the natural 4 bases, creating synthetic base pairs that function in living cells. Organisms incorporating these unnatural base pairs can produce proteins containing non-standard amino acids with novel properties. This work tests fundamental assumptions about the nature of genetic information and could enable new biotechnologies. The possibility of life with alternative biochemistries has implications for the search for life beyond Earth and for understanding the limits of biological possibility. Astrobiology, the study of life in the universe, draws heavily on genetics and molecular biology. Understanding how life originated on Earth and what chemical and physical conditions are required for life informs the search for life elsewhere. The discovery of extremophile organisms thriving in conditions once thought incompatible with life, from deep-sea hydrothermal vents to acidic hot springs, has expanded our conception of where life might exist. These discoveries rely on genetic analysis to understand how organisms adapt to extreme conditions and to identify previously unknown forms of life. As you drift deeper into relaxation, let your mind wander through these realms of genetic knowledge. Consider the elegant simplicity of base pairing that enables DNA replication, the intricate regulatory networks that control when and where genes are expressed, the constant molecular vigilance that maintains genome integrity, and the evolutionary forces that have shaped the genetic diversity of life. From the chemical bonds between nucleotides to the inheritance patterns that connect generations, from single-cell organisms to complex multicellular life, genetics provides a unifying framework for understanding biology. Your own genome is unique, a singular combination of genetic variants inherited from your ancestors and shaped by new mutations, yet also deeply connected to all humanity and all life through shared evolutionary history. The same fundamental processes of replication, transcription, and translation occur in your cells as in all living things, demonstrating the profound unity underlying biological diversity. Molecular evolution studies how DNA sequences change over time and uses these changes to infer evolutionary relationships between organisms. The molecular clock hypothesis proposes that mutations accumulate at relatively constant rates, allowing scientists to estimate divergence times between species by counting genetic differences. Different genes evolve at different rates, depending on how strong selective pressure acts on them. Genes encoding essential proteins with critical functions tend to evolve slowly because most mutations are harmful and eliminated by natural selection. Genes with less constrained functions, or non-coding DNA regions, may accumulate changes more rapidly. Comparing rates of synonymous substitutions, changes that do not alter amino acid sequences, with non-synonymous substitutions, changes that do alter amino acids, helps determine whether natural selection has been acting on a gene. When non-synonymous substitutions occur more frequently than synonymous ones, positive selection may be driving evolutionary change. When synonymous substitutions predominate, purifying selection is likely removing harmful variants. Balanced polymorphism occurs when natural selection maintains multiple alleles in a population, because heterozygotes have advantages over either homozygote. The sickle cell allele provides a classic example, as individuals with one sickle cell allele and one normal allele have resistance to malaria without suffering from sickle cell disease, which affects individuals with two sickle cell alleles. In regions where malaria is common, this heterozygote advantage maintains both alleles in the population, despite the serious health consequences of the homozygous sickle cell genotype. Genetic load refers to the reduction in population fitness due to the presence of deleterious alleles. Every individual carries many mildly harmful mutations, most in heterozygous state, where their effects are masked by normal alleles. Mutation load, the portion of genetic load due to recurrent mutation, is balanced by selection against harmful variants. Inbreeding depression, reduced fitness in offspring from closely related parents, results from increased homozygosity exposing recessive deleterious alleles that were hidden in heterozygous state. This phenomenon explains why most organisms have evolved mechanisms to avoid inbreeding. The major histocompatibility complex, or MHC, is a cluster of genes encoding proteins that present foreign antigens to immune cells, enabling the immune system to distinguish self from non-self. MHC genes are extraordinarily diverse, with hundreds of different alleles at some loci in human populations. This exceptional diversity is maintained by balancing selection, as rare MHC alleles may provide advantages against new pathogens. Some studies suggest that MHC diversity influences mate choice, with preferences for partners with dissimilar MHC genes, possibly evolved to increase offspring MHC diversity and immune competence. Human leukocyte antigen, or HLA, genes are the human version of MHC genes and play crucial roles in immune function and transplant compatibility. HLA matching between donors and recipients is critical for successful organ and bone marrow transplants, because mismatched HLA proteins can trigger immune rejection. Finding compatible donors can be challenging due to HLA diversity, particularly for individuals from minority ethnic groups who are underrepresented in donor registries. Population genetics models describe how allele frequencies change over time under various evolutionary forces. The Hardy-Weinberg equilibrium provides a null model for populations not experiencing evolution, stating that allele frequencies remain constant across generations in the absence of mutation. Selection, genetic drift, gene flow, and non-random mating. Real populations deviate from Hardy-Weinberg equilibrium in various ways, and measuring these deviations helps identify evolutionary forces at work. Genetic bottlenecks occur when population size dramatically decreases, reducing genetic diversity as rare alleles are lost. The founding population for any subsequent expansion carries only a subset of the original genetic variation. Bottlenecks can explain why some populations have less genetic diversity or unusual allele frequencies compared to larger populations. Human populations that migrated out of Africa experienced bottlenecks, resulting in reduced genetic diversity compared to African populations. Similarly, endangered species with small population sizes face genetic consequences of bottlenecks that can affect their long-term viability. Gene flow, the movement of alleles between populations through migration and interbreeding, counteracts the divergence that would occur through genetic drift and local adaptation. Gene flow can introduce beneficial alleles into populations where they were absent, or conversely, it can swamp local adaptations by introducing alleles that are maladapted to local conditions. The balance between gene flow and local selection determines whether populations maintain distinct characteristics or homogenize genetically. In humans, historical patterns of migration and gene flow have shaped the geographic distribution of genetic variants. Isolation by distance, where genetic similarity decreases with geographic distance, occurs because gene flow is more likely between nearby populations than distant ones. Coalescent theory traces genetic lineages backward in time to their most recent common ancestors, providing a framework for understanding genetic relationships between individuals and populations. Every pair of DNA sequences, whether from different individuals or different populations, ultimately traces back to a common ancestral sequence through a series of mutational changes and reproductive events. The time to coalescence, when lineages merge in their shared ancestor, depends on population size and demographic history. Mitochondrial Eve, the most recent common ancestor of all human mitochondrial DNA, lived approximately 150,000 to 200,000 years ago in Africa. This does not mean she was the only woman alive at that time, but rather that her mitochondrial lineage is the only one that has survived to the present through an unbroken chain of daughters. Similarly, Y chromosome Adam, the most recent common ancestor of all human Y chromosomes, lived approximately 200,000 to 300,000 years ago. These genetic ancestors lived at different times and were not a couple, demonstrating the independent inheritance of different genomic regions. Phylogenetics uses genetic data to infer evolutionary relationships and construct evolutionary trees showing how species or populations are related. Different methods exist for tree construction, including maximum parsimony, which seeks trees requiring the fewest evolutionary changes, maximum likelihood, which finds trees that make observed data most probable under specific evolutionary models, and Bayesian inference, which incorporates prior knowledge and quantifies uncertainty in evolutionary relationships. Molecular phylogenies have resolved many long-standing questions about relationships between organisms and have sometimes revealed surprising connections not evident from morphology alone. Horizontal gene transfer between bacteria enables rapid spread of genes conferring antibiotic resistance, creating serious public health challenges. Resistance genes can spread between different bacterial species sharing the same environment, such as the human gut. Plasmids, small circular DNA molecules separate from the bacterial chromosome, often carry antibiotic resistance genes and can transfer between bacteria through conjugation. Transposons, mobile genetic elements that can insert themselves into different locations, can move between plasmids and chromosomes, facilitating horizontal gene transfer. The spread of antibiotic resistance through horizontal gene transfer highlights the evolutionary consequences of antibiotic use and the importance of judicious antibiotic stewardship. Metagenomic sequences all DNA in environmental samples without culturing individual organisms, revealing the genetic diversity of microbial communities. This approach has discovered vast numbers of previously unknown microorganisms and genes, greatly expanding our understanding of microbial diversity. The human gut microbiome, soil microbial communities, ocean microorganisms, and many other microbial ecosystems have been characterized through metagenomic studies. These studies reveal complex communities with extensive genetic diversity and metabolic capabilities. Functional metagenomics goes beyond identifying what organisms are present to understanding what metabolic activities they perform, connecting genes to their functions in natural environments. The concept of the holobiont recognizes that multicellular organisms and their associated microorganisms function as integrated units. From this perspective, an organism's phenotype results from both its own genes and the collective genes of its microbiome. The hologenome, the combined genetic information of host and microbiome, provides a broader view of heredity and adaptation. Some traits previously attributed solely to host genetics may actually involve contributions from microbial genes. This perspective has implications for understanding health, disease, and evolution. Epigenetic inheritance between generations has been documented in various organisms. In plants, epigenetic marks can persist through meiosis and affect offspring phenotypes, providing a mechanism for rapid adaptation to environmental changes. In mammals, most epigenetic marks are erased and reset during gamete formation and early development, but some escape reprogramming and can be inherited. The extent of epigenetic inheritance in humans and its significance for evolution and disease remain debated and actively researched. Environmental effects on epigenetics have been demonstrated in various contexts. Maternal nutrition during pregnancy can affect offspring DNA methylation patterns and subsequent health risks. Exposure to toxins or stress can alter epigenetic marks. Some studies suggest these environmentally induced epigenetic changes can persist into subsequent generations. Though the mechanisms and prevalence of transgenerational epigenetic inheritance in humans remain unclear. These findings challenge simple nature versus nurture dichotomies and highlight complex interactions between genes and environment across generations. Phenotypic plasticity, the ability of a single genotype to produce different phenotypes in different environments, is ubiquitous in nature. Some plastic responses involve epigenetic mechanisms that alter gene expression without changing DNA sequence. Developmental plasticity can lead to permanent differences between individuals with identical genes raised in different conditions. Acclimation and acclimatization involve physiological adjustments to environmental changes, sometimes involving altered gene expression. The genetic basis of plasticity itself can evolve, with selection favoring genotypes that respond appropriately to environmental variation. Canalization refers to the developmental buffering that makes phenotypes relatively insensitive to genetic or environmental variation. Highly canalized traits remain consistent despite perturbations, while less canalized traits are more variable. Canalization evolves through natural selection, because individuals whose development is robust to perturbations may have higher fitness than those whose development is disrupted by normal variation. However, excessive canalization might prevent adaptive responses to environmental changes, creating trade-offs between robustness and flexibility. Systems biology approaches integrate data on genes, proteins, metabolites, and cellular processes to build comprehensive models of biological systems. Rather than studying individual genes in isolation, systems biology examines networks of interacting components and emergent properties that arise from these interactions. Gene regulatory networks describe how transcription factors control expression of target genes, which may themselves encode transcription factors, creating complex feedback loops. Metabolic networks map biochemical reactions and their regulation. Protein interaction networks chart physical associations between proteins. These networks exhibit properties like robustness to perturbations, modularity with distinct functional units, and scale-free architecture, where a few highly connected hubs interact with many other nodes. Understanding network properties helps explain how biological systems function and how they fail in disease. Personalized medicine tailors treatments to individual characteristics, including genetic makeup. Genetic variants affecting drug metabolism, drug targets, or disease mechanisms can guide treatment selection and dosing. Tumor genetic profiling identifies mutations driving specific cancers, enabling selection of targeted therapies likely to be effective for that tumor. Polygenic risk scores combine information from many genetic variants to estimate individual disease risks, though their predictive power varies by disease and population. Implementation of personalized medicine faces challenges including cost, interpretation complexity, health disparities, and integration into clinical workflows. The promise of personalized medicine must be balanced against potential risks of over-testing, incidental findings, and psychological impacts of genetic risk information. Nutrigenomics studies interactions between nutrition and genes, examining how genetic variants affect nutrient metabolism and dietary needs, and how nutrients influence gene expression. Some genetic variants affect vitamin requirements, caffeine metabolism, taste perception, or food sensitivities. Dietary components can influence gene expression through effects on transcription factors and epigenetic modifications. While nutrigenomics promises personalized nutrition recommendations, the field is still developing, and many claim gene-diet interactions lack robust evidence. Genetic ancestry testing uses DNA variants to infer geographic origins of an individual's ancestors. These tests compare an individual's genetic markers with reference populations from different regions, estimating ancestry proportions. While these tests can provide general insights into geographic ancestry, they have limitations and should be interpreted cautiously. Ancestry is a continuum rather than discrete categories. Reference populations may not capture full diversity of human variation, and results can vary between companies using different methods and databases. Genetic ancestry differs from cultural or legal definitions of identity and should not be conflated with race, which is a social rather than biological construct. Forensic genetics applies genetic analysis to legal questions, most commonly using DNA profiling for identification. DNA from crime scenes can be compared with suspect samples or searched against databases of convicted offenders. Probabilistic genotyping software helps interpret complex mixtures of DNA from multiple contributors. Mitochondrial DNA, and why chromosome analysis provide information when nuclear DNA is degraded or limited. Forensic genetic genealogy, using DNA matches in consumer databases to identify suspects through relatives, has solved cold cases, but raises privacy concerns about use of genetic information for purposes beyond those originally consented to by database participants. Genetics of behavior is complex and controversial. While genes influence behavioral traits, most behaviors result from many genes with small effects plus environmental factors. Twin studies suggest genetic contributions to personality traits, intelligence, mental health conditions, and substance abuse. But heritability estimates do not imply immutability or diminish importance of environment. Claims about genetic bases of complex behaviors should be evaluated critically, as history shows how genetic determinism has been misused to justify discrimination. Understanding genetic influences on behavior requires sophisticated approaches, recognizing gene-environment interactions and avoiding oversimplification. Neurogenetics studies genetic influences on brain development and function. Many neurodevelopmental and psychiatric disorders have genetic components. Autism spectrum disorders involve hundreds of genes affecting brain development and connectivity. Schizophrenia and bipolar disorder show strong heritability with numerous genetic risk factors identified. Depression and anxiety disorders have both genetic and environmental contributions. Neurodegenerative diseases like Alzheimer's disease and Parkinson's disease involve genetic and environmental risk factors, with some cases caused by single gene mutations, but most being multifactorial. Understanding the genetics of brain disorders may eventually lead to better treatments and prevention strategies. Genetic counseling helps individuals and families understand genetic conditions and make informed decisions. Counselors explain test results, discuss risks and options, and provide psychological support. Genetic counseling is non-directive, supporting patient autonomy rather than advising specific courses of action. Situations where genetic counseling may be valuable include family history of genetic conditions, abnormal prenatal screening results, recurrent pregnancy loss, consanguineous marriages, and consideration of genetic testing. Prenatal genetic testing includes screening tests that assess risk and diagnostic tests that provide definitive answers. Maternal blood tests screen for chromosomal abnormalities and some single-gene disorders. Ultrasound examines fetal anatomy for structural abnormalities. Diagnostic tests like amniocentesis or chorionic villus sampling analyze fetal cells obtained invasively, providing detailed genetic information but carrying small procedural risks. Non-invasive prenatal testing analyzes fetal DNA fragments in maternal blood, providing high accuracy for detecting common chromosomal abnormalities without procedural risks. Parents must decide whether to undergo testing and how to use the information obtained. Decisions involving medical, ethical, and personal considerations. Preimplantation. Genetic testing analyzes embryos created through in vitro fertilization before transfer to the uterus. This allows selection of embryos without specific genetic conditions or with desired characteristics like HLA matching to an existing sick child who needs a stem cell donor. While preimplantation genetic testing can help families avoid serious genetic diseases, it raises ethical concerns about embryo selection, particularly regarding potential applications for non-medical trait selection. Current professional guidelines limit preimplantation genetic testing to serious medical conditions. Gene patents, granting exclusive rights to use genetic sequences, were once common, but have become more restricted following legal decisions, holding that naturally occurring DNA sequences are not patentable. Patents on genes had complicated genetic testing by requiring licenses and increasing costs. The U.S. Supreme Court's 2013 decision in Association for Molecular Pathology v. Myriad Genetics held that isolated DNA is not patentable, but complementary DNA, which is synthetically created, remains patentable. This decision improved access to genetic testing. However, patents on methods of genetic analysis and gene therapy technologies continue, balancing innovation incentives with access to genetic information and treatments. Genetic exceptionalism treats genetic information as qualitatively different from other medical information, warranting special protections. Arguments for exceptionalism note that genetic information is immutable, predictive of future conditions, and reveals information about relatives. However, critics argue genetic information is not fundamentally different from other medical information that also has predictive value and affects families. Whether genetic information deserves special treatment remains debated in medical ethics and health policy. Consumer genomics companies offer genetic testing directly to consumers without physician involvement. Services include ancestry analysis, health risk assessment, trait reports, and carrier screening. While these services democratize access to genetic information, concerns exist about result accuracy, psychological impacts of unexpected findings, privacy of genetic data, and adequacy of support for interpreting results. Regulatory oversight varies by jurisdiction, and quality standards are still evolving. Consumers should carefully evaluate companies and understand limitations before testing. Genetic discrimination, unfair treatment based on genetic information, is a significant concern. The U.S. Genetic Information Non-Discrimination Act prohibits discrimination in health insurance and employment, but does not cover life insurance, disability insurance, or long-term care insurance. Some people avoid genetic testing fearing discrimination despite potential medical benefits. International regulations vary widely. Balancing protection against discrimination with legitimate uses of genetic information remains challenging. Gene doping, using genetic technologies to enhance athletic performance, is prohibited in sports but difficult to detect. Potential approaches include gene therapy to increase muscle mass or enhance oxygen carrying capacity. While no confirmed cases exist, the possibility concerns anti-doping authorities. Detection methods are being developed but face technical challenges. The ethics of genetic enhancement in sports parallels broader debates about enhancement versus therapy in genetic medicine. Designer babies, children whose genetic characteristics have been deliberately modified or selected, represent a hypothetical and controversial application of genetic technology. Current technologies like preimplantation genetic testing allow selection among embryos but not modification. Gene editing could potentially allow more direct modification. While correcting disease-causing mutations may be acceptable, enhancement of traits like intelligence or appearance raises serious ethical concerns about inequality, changing human nature, and parents' rights versus children's autonomy. Most ethicists and scientists oppose genetic enhancement of human embryos with current knowledge and technology. Genetic enhancement more broadly raises questions about using genetic technologies to augment capabilities beyond normal function. While some enhancements like correcting vision problems are widely accepted, others like increasing intelligence or physical abilities through genetic modification are controversial. Concerns include safety, fairness, access, social pressure, and effects on human diversity and identity. Some argue enhancement is natural extension of medicine's goals, while others see fundamental distinctions between treating disease and enhancing normal function. These debates will intensify as genetic technologies advance. Genetic identity and privacy are increasingly important as more people undergo genetic testing and as genetic data is shared for research. Genetic information is inherently identifying and difficult to anonymize. Relatives share genetic information, so one person's genetic data reveals information about family members who may not have consented. Balancing research benefits requiring data sharing against privacy rights is challenging. Regulations like GDPR in Europe provide some protections, but international harmonization is lacking. Individuals should understand how their genetic data will be used and protected before participating in genetic testing or research. Genetic literacy, understanding basic genetic concepts and their implications, is essential for informed decision-making about genetic testing and participation in policy debates. However, studies show many people lack basic genetic knowledge. Improving genetic education in schools and healthcare settings could help people understand genetic information and resist genetic determinism and discrimination. Healthcare providers need genetics education to effectively use genetic information in practice. Public engagement in genetics policy decisions requires accessible, accurate information about genetic technologies and their implications. As you continue drifting into deeper relaxation, let these concepts about DNA and genetics wash over you like gentle waves. The intricate mechanisms of heredity, the complex interplay between genes and environment, the unity of life revealed through shared genetic codes, the power of modern genetics to transform medicine, and the profound ethical questions raised by genetic technologies all invite contemplation. Your unique genome connects you to all humanity through shared ancestry, while making you distinct through your particular combination of variants. Genetic processes operating over billions of years have created Earth's biological diversity, while maintaining fundamental continuities in the molecular machinery of life. From the spiral staircase of the double helix to the three-dimensional architecture of chromosomes, from single nucleotide changes to massive chromosomal rearrangements, from silent mutations to dramatic phenotypic effects, genetics encompasses scales from molecules to populations. Time spans from cell cycles to evolutionary epochs. Understanding genetics illuminates who we are, where we came from, and possibilities for the future while raising important questions about responsibility and wisdom in applying genetic knowledge. The future of genetics holds exciting possibilities and challenges. Continued advances in sequencing technology may make routine genomic sequencing part of standard healthcare. Improved understanding of gene function and regulation could enable more effective treatments for genetic diseases. Gene editing technologies may become safer and more precise, expanding therapeutic applications. Artificial intelligence and machine learning may help interpret complex genetic data and predict gene function. Synthetic biology may create new organisms with beneficial capabilities. These advances promise improvements in medicine, agriculture, environmental remediation, and basic understanding of life. However, they also require careful consideration of ethical implications, equitable access, environmental impacts, and unintended consequences. The responsible development and application of genetic technologies will require ongoing dialogue between scientists, ethicists, policymakers, and the public. Education about genetics will become increasingly important as genetic information becomes more integrated into everyday life. Understanding how genes work, how genetic variation arises, how traits are inherited, and how genes interact with environment will help people make informed decisions about genetic testing. interpret genetic information appropriately, and participate in societal discussions about genetic technologies. Combating genetic determinism, the misconception that genes rigidly determine all traits, requires understanding that most characteristics result from complex interactions between many genes and environmental factors. The genetic instructions that have enabled life to persist and diversify over billions of years continue their work in your cells right now, maintaining the countless processes that support your health and well-being. Sweet dreams, and may you wake refreshed and restored.